Welcome everybody on a windy Sydney night. Thank you for coming along. My name is Ian Young. I'm the Dean of Science at the Faculty of Science in Sydney University. I'd like to thank you all for taking the time to come here tonight to what will be a very illuminating talk. Thank you very much. Before we begin, can I just do an acknowledgement of country? I would like to acknowledge and pay respect to the traditional owners of the land in which we meet, the Gadigal people of the Aurora Nation. It is upon their ancestral lands that the University of Sydney is built. As we share our knowledge, teaching, learning and research practices within this university, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. Ironically, I can hardly read. My think is of the lack of light, so as I bend down, please bear with me. It's a great pleasure to introduce Associate Professor Liz New and Dr. Alice Motion from the School of Chemistry. Liz has studied and worked in Australia, the UK and the US, and is currently Associate Professor and Westpac Research Fellow at the University of Sydney. She and her team of 18 study light-emitting molecules and their applications, including fluorescent tracking of molecules in the body to better understand diseases. Her awards for research and teaching, including the 2018 Eureka 3M Emerging Leader Prize and the 2016 RACI Chemistry Educator of the Year. Alice Motion has studied and worked in the UK, Switzerland and Australia and is currently lecturer in chemical education and outreach at the University of Sydney, again in the School of Chemistry. Alice is an open source scientist and director of Breaking Good, a project that empowers young people and citizens to make molecules that matter. Her research is centred on ways to connect people with science. Alice was a finalist in the Google Impact Challenge, Australia 2018, has presented a weekly science segment on FBI Radio since 2015, and is the co-host of the ABC Science podcast, Dear Science. So it's my great pleasure to actually introduce Liz and Alice to present what will be a very illuminating talk.
Hello, I'm Alice, and this is Liz. As Ian very kindly introduced both of us, we are both from the School of Chemistry. We are friends and colleagues who work together on a number of things, and this year, for the first time, we are academic co-parents, and we're going to share some of Liz's research that she does in her research group today, and take you on, hopefully, an exciting journey looking at light in a different way. So we thought it was best to start a talk about light in the dark. And maybe just take a moment, even though we can see some light in this room, to imagine the way that we would experience the world if we didn't have any light. How would we recognize the people that we love? How would we see the beauty of nature all around us? And how would we appreciate things if we weren't able to see the light that hits our eyes and illuminates the world around us? But where did light begin? When was the first light? Oops. So for the first light, we have to think back to 1964. Light, or 1964, I'm reliably informed, but a discovery by Zayas and Wilson when they were listening to their telescope was rather extraordinary. What we're hearing now is the first light we were able to detect in the universe. the most important source of light for us here on Earth, and in fact the most important source of heat, of energy, for pretty much everything that's living on the planet, is our nearest star, the sun. And when we, feel this sunrise, we feel light that's come from the surface of the sun about 8 minutes and 20 seconds ago. But actually, with each sunrise, we're feeling light that has its origin around 100,000 years ago. Because this light begins at the center of the sun, and it takes an awful long time for it to reach the surface. So you can think about that. Next time you see the sunrise, next time you feel that light on its face, you could be feeling light that started at least 100,000 years ago. So that's pretty amazing. Now, the sun also sets something really important for all of us. It sets our circadian rhythm. As we feel sunlight, as it hits the carbon, all of the molecules that make our body, make us who we are, the sun actually starts this circadian rhythm that most animals and plants live according to on the planet. We all have this kind of 24-hour cycle. Now, as humans, we are particularly interested in a subsection of this light. But there is a whole range of light that really comes from radio waves, which are really long. They can be as long as planets, actually. But when we hear radio waves, they're typically the ones that we listen to our favorite radio station with, typically kind of the length of buildings. And they go all the way down to gamma rays at the other end, which can be smaller than a hydrogen atom. So there's this whole range of light that we really can think about as being part of the electromagnetic spectrum. And we as humans are a little bit obsessed with this tiny slice of visible light here in the middle. Now, this scale is not to scale. This is a very tiny window of light that we think of as being important. And there's nothing particularly important about it, apart from the fact that it's the light that we can see. And why is that? Well, it could be for a number of reasons. Part of the reason that people think that we can see this small window of light from around 400 to 700 nanometers, which is about the size of a bacteria, or about 1% of the size of a human hair, is because the first eyes are thought to have evolved underwater. And underwater, this region of light can propagate, it can travel through water well. So that's potentially why. And when we think about the human body and we think about the molecules that make us who we are, it's this range of light that stimulates our eyes to start this vision process. Electromagnetic waves that are longer don't have enough energy to kickstart this process, and those that are shorter have too much energy, and they can start to damage our body. We know that UV light can damage our skin, for example, or cause cancer. Now, all of these types of electromagnetic radiation travel at the speed limit of the universe, which is 300,000 kilometers per second. And most people agree that this is the fastest that anything can travel in our universe. Most people, apart from Terry Pratchett. So Terry Pratchett says that, or he said in one of his books, Reaper Man, light thinks it travels faster than anything, but it is wrong. No matter how fast light travels, it finds the darkness has always got there first and is waiting for it. So now, unfortunately, we're switching to something a little bit dark in a different way. We're going to go to a crime scene. So for those of you who like watching gory TV programs and followed the progress of all of the CSIs, I think there's one for every state or country, state in America rather, may be familiar with the technique that these detectives use when they go into a crime scene. The crime scene might appear clean, but when you switch off the lights, the detectives spray a certain substance, they see something that lights up under UV light. And we're going to show you an example of what's happening in this chemical reaction now and how we, as chemists, can make light. So I'll just put the lab coat on, because we're getting serious. And I have two solutions here, which I should show you quickly before it gets too dark. This one is dark. It's sort of black in color. This one is clear and colorless, I promise. I'm not fibbing. I'll put the lab coat on just in case everything blows up. I think you should be okay on the third row. But... So we go to the take these two solutions. One is clear and colorless. One is black. I'm going to mix them together. I'm going to try not to spill it on the table. And I'd like you to see what happens. So we have a reaction happening between some chemicals that I have prepared earlier. In fact, I haven't prepared earlier. Lovely Elizabeth and Mary from the School of Chemistry have prepared them for us, so thank you very much. And what we have in this mixture, if I can find the clicker, because it's very dark down here. What we have in this mixture is a mixture of hydrogen peroxide in one of our solutions, which is the same stuff that is sometimes taken in a very small quantity as in hair dye to dye hair blonde. And we also have a molecule called luminol. So this is the luminol is the big structure here with lots of N's in, lots of nitrogens in. And this reacts with hydrogen peroxide in the presence of a catalyst, which in this case is copper. It's the metal copper. And then as we react those two substances together, what happens is that we add some energy. There's a chemical reaction where the electrons in this molecule get really excited. And they jump up into a higher energy state. Then they want to lose this energy, and those electrons fall down to a lower level, and they emit this energy in the form of light. Now, I mentioned here that we have a catalyst of copper. So if we go back to our crime scene, does anybody know what the catalyst might be in human blood? A metal that's in human blood. Yeah, it's iron. So it's the iron in hemoglobin that acts as a catalyst for this reaction in a crime scene. And that's why we can see those blood stains long after they've been cleaned up if you spray some luminol on them and then shine a UV light. So we're going to switch tacks from a crime scene to a rave. So Liz and I were at a rave last night, and we were wearing glow sticks. And the chemical reaction that happens in a glow stick, if any of you have ever worn one, when you snap this glow stick, it's a very similar reaction that's happening. There is a chemical process where we have some excitement of these electrons that are raised in energy. They go to a higher energy state, and this is then released as it falls back to their lower energy in the form of light. It's a little bit like when you arrive at a rave. You might be feeling quite low energy, but when you hear the music, you get some energy from the music and you get into an excited state and you start dancing. And then at the end of the night, when the music stops, you lose that energy and fall back down to the ground state. And we're going to show you this with some juggling balls. So if we think about this table as being the ground state, and I lift this juggling ball up to an excited state, and then I drop it, it releases this energy and falls back down to the ground state.
And it's releasing its energy in the form of sound, in this case. So what happens if I raise mine to a different level?
So let's see if we can hear a difference in the sound. Let's see if this works. Yeah. So this one's much louder. It's falling from a higher energy level, and it's releasing more energy in the form of sound because it's fallen further. Now, in these glow sticks, because the different chemicals that are mixed in here, we can change the level to which that electron is excited. And if an electron is excited to a higher energy level, the light that's given off is of a higher energy. So it means it's more like a violet color. If we excite it to a lower energy level, the light that's given off is a lower energy, so it's more like red. And we can see this. We're going to show you a similar slide to this for each of these processes. We start with an electron that's in its ground state. We put some energy into this system, so it's now gone to its excited state. And then this energy is released in the form of light. Now, for those of you who've been lucky enough to maybe visit the North Island of New Zealand, you may have seen this phenomena that we've just shown as a chemical reaction that happens in a natural setting. So I'm just going to play you a short video now of some beautiful glowworms. I'm not going to play you a short video, I'm going to tell you about it. So, thanks, Liz. So each of these glowworms is giving off energy in the form of light. And it's a cold light. Thankfully, these glowworms don't get too hot. If you've ever touched a light bulb at home, you can burn your hand. Don't hold your hand next to a light bulb. But this light is a cold light. And we can see this in other species too. So here we have a firefly, which is giving off a different color of light. So if you see those pictures on the right, we have a picture of a firefly in daylight, and we can't really see anything too exciting. But when we switch off the lights, we can see that there's this green fluorescent glow from the abdomen of the firefly. And in this system, it's still a chemical reaction that is raising the energy of these electrons, but it's a chemical reaction that's happening in a biological system. So it's a biochemical reaction. And again, this energy is released in the form of light. So something that you may be more familiar with, especially if there's anyone who's revising for exams at the moment. I'm glad that you're here and not the library, but do hurry back. These are highlighters, and you may have used them to take notes or to record things that are important. And highlighters have an interesting property in that some of them are fluorescent. Now, Liz's group have been busy in the research lab all afternoon, drawing some pictures for us using highlighters. That's not what they normally do. But we're going to show this to you now. So if we have a look at an image, which is a little bit bright, an image of a cat. It's been drawn on a filter paper. And we can see that there's a blue highlighter to outline this cat. The skeleton of the cat is drawn in a yellow highlighter. The paw prints of the cat are in a green highlighter. There's a little orange mouse, and it has pink fluorescent paw prints. And now if we have a look at this underneath a UV light, you might notice that the blue has disappeared. And that's because all of the other highlighters are fluorescent, but the blue highlighter isn't. And this is a bit strange because if you take a green highlighter and you separate out the colours that make it green, it contains yellow highlighter, which is fluorescent, and blue highlighter, which isn't. So there's a bit of trivia for you. Blue highlighters aren't fluorescent, but all of the other ones are.
And look, we can even see it here because the green paw prints look really yellow under UV light.
So what's happening in these systems with the highlighters, it's something very similar to the processes that we've already shown you. We're seeing an example of a system where when we put energy into this system, we see that this light is emitted in the form of a very bright fluorescent light. So in this case, in the previous cases, we've seen that we've put energy into the system in the form of a chemical reaction. Here, you saw that when we shone the UV light onto this drawing, we could see that those molecules inside those highlighter pens, some of the electrons receive energy, which is then emitted in the form of light, and that's why we see this fluorescence. And UV light is particularly good at this for lots of systems. It's the right energy for us to put energy into lots of systems. And that's why it's used so commonly in fluorescence. So for example, I borrowed some money off Liz recently. I was going on a trip to Europe, and she very kindly has lent me 50 euros. And she doesn't really trust people. She's a scientist, so she likes to test everything. So she's going to check in front of all of you that this is an authentic 50 euro.
Oh, it's a lot more scrunched up than when I lent it to you.
It's a different one. I spent the one you gave me.
Okay, so let's first just look at it under regular light and see. Oh, sorry.
Yep.
See what it looks like. Yep, that looks like a euro, but I'm still not sure. You could have just photocopied it. So let's see what we see under UV light.
Could never get anything past a scientist. They always want to do some form of experiment. So let's see what happens here. So we can see that there's some yellow fluorescence, but I know you're looking for something different.
Oh, yes, I'm looking for that red fluorescence, that really nice stripe.
Can everyone see that? Trust me?
So I think we can safely say that that's a genuine euro. And one really interesting thing about that red fluorescence is that it comes from an element called europium. And that element was named after Europe. So it's quite fitting that it is what is used to give this beautiful color on this euro.
Now, the good news is that Europe might have its UV forgery techniques, but in Australia we have a much more sophisticated system in addition to this. And this is that deliberately misspell words on our notes so that forgers have to be very careful when they're recreating them. So we're going to change track now. We've talked about fluorescence in terms of molecules that are in terms of reactions in living systems, in terms of reactions in chemical systems. We can also design chemicals and synthesize them that have fluorescent properties. And this is one of the most famous examples of a fluorescent compound. This is Fluorescein. You can see it fluorescing here on the UV lamp. And this molecule was synthesized at the end of about the 1870s, I think 1871, by Adolf von Baeyer, who was a German chemist and who later won the Nobel Prize for some of his work on dyes and other inventions. And this molecule is really interesting because it has this particularly potent fluorescence, and it's been used for several applications. In World War II, fighter pilots were actually given small quantities of fluorescein so that in the unfortunate event, and unfortunately frequent event for all sides, sometimes they were ejected from their planes and they might end up in the ocean. And they could open this vial of fluorescein, place it in the ocean, and even though fluorescein fluoresces at its very best under UV light, there's enough UV light in the sun for it to fluoresce green in the ocean and for them to find some of those fighter pilots. Now, for a more cheerful application, does anyone recognize this river? Chicago, yes. So can anyone guess what time of the year we might be celebrating? St. Patrick's Day. So fluorescein was used in the 60s as the first dye to light up the Chicago River. And it looked quite similar to this because, again, there was enough UV light from the sun to cause the fluorescein to fluoresce and to give this green color. In 1966, people got a little bit unhappy about the use of fluorescein in the river, and so it's now been switched to a vegetable-based dye, which is thought to be less harmful for the life of species living in the river. But nonetheless, it can produce this really quite magnificent green for people to celebrate St. Patrick. Now, it's also been used in other applications for rivers, and for this one, we're going to do a bit of a role play. So you've got to imagine that we are about 6 years after Adolf von Bayer's discovery in 1877. We're both scientists. We're based in Europe. Liz is in Austria. I'm in Germany. And Liz wants to check to see whether the Danube is connected to the Rhine.
So I've heard that would be really useful for shipping, because imagine if we could ship things from one end of Europe to the other by using one river system rather than having to stop and carry stuff overland into the Rhine.
And so how would you do this? Well, one idea would be to put something in the river and to see if it ends up in Germany. Now, it might be a bit hard to detect with a stick or a piece of log because it could get lost along the way. We could put it in dye, but even better, we can use a dye that fluoresces. And this is what some people did back in 1877. So they put some fluorescein in the river.
So they put fluorescein at the end of the Danube, and a few days later did some tests in the Rhine.
And it turned up in the Rhine. Now, this was something that told us a lot about the river's geography, because actually it was a sunken river. It was a river that was deep underneath the ground, and that was connecting to the Rhine in this way. And the reason they use fluorescein rather than another dye is because of just how potent it is, and we might be able to show you that with some samples that Liz prepared earlier.
So we saw this sample of fluorescein before, and you can almost see the fluorescence of it just under normal light. But I then made this sample, which was 1,000,000 times less concentrated. And now we can't see it at all with our eye. We can't see the color of the fluorescein. But if we have a look instead at the fluorescence, we can see the fluorescence. And that's because fluorescence is a far more sensitive method than just absorbance, than just looking with our eye at the light that we see absorbed. And this is the reason that it's much more useful to use a fluorescent dye, because we need it in much lower quantities than we would need to use a colored dye.
So this is one of the ways that fluorescein's been used. We're going to talk about some other applications a little bit later in the talk. But from celebrating St. Patrick's Day to understanding the geography of rivers in Europe, it's a pretty useful molecule. So we've talked about some compounds that we can synthesize, like fluorescein, and rely on and design them so that they have fluorescent properties. But there are a lot of molecules that occur naturally that have fluorescent properties. And this is a sample here of turmeric. So we have some turmeric in a powder form from the supermarket. Many of you will have heard lots of things about the health benefits of turmeric. It's because of a molecule called curcumine that's present in turmeric, and is a fluorescent molecule. Some of our friends wrote an article for The Conversation recently and say, although turmeric is delicious and very tasty, if you wanted to eat enough turmeric to get the right quantity, a medicinal quantity of curcumine in your body, you'd have to eat 30 grams a day, which is quite a lot of turmeric. And it's actually not very bioavailable. It doesn't dissolve very easily in the body. So you'd probably have to eat closer to 100 grams of turmeric if you wanted to reach those medicinal properties. And I don't know about you, I really like curry, but 100 grams of turmeric a day is a little bit too much. So we can now have a look at the fluorescent properties of this molecule. So those of you who are familiar with turmeric, it's this kind of orangey, brown-coloured powder, and it fluoresces when it's dissolved in ethanol. Fluoresces yellow. Now, there are other molecules that are also fluorescent, and one of which is tonic water. So I'm going to open this now and hope it doesn't fizz everywhere. So tonic water is fluorescent because of the presence of one of its most famous components, which is quinine. I could see you mouthing it there, sir. So quinine is a molecule that has a rich history in the treatment of malaria. It's extracted from a bark. People used to drink gin and tonic to try and protect themselves from malaria, although that might have been a delicious way to enjoy one's evenings. I did a calculation before, and if you wanted to drink enough tonic water to have a medicinal quantity of quinine, there's never more than 83 milligrams of quinine per litre of tonic water, and you need 1.5 grams of quinine if you want to have a medicinal dose. So I think that's about 18 litres of tonic water. Gin is optional, but it's still not very sustainable. So let's have a look and see if this fluoresces. So you can see that fluoresces very nicely, and it's a way to check that people have genuinely given you tonic water if you're drinking that. So carry your UV light with you at all times. And then perhaps an Aussie favourite, so Vegemite. So Vegemite also fluoresces. While we're just preparing this, I'll tell you a little story. Before I came to Australia and saw the light, I used to be a fan of Marmite, but I now recognise that this is better. And when I tried to introduce Marmite to my laboratory when I worked in Switzerland, I brought some in to prepare a breakfast for my French co-workers. And one of my co-workers just spat out her bread instantly. My boss was much more polite, so he stood there very quietly, chewed every piece of the bread with some Marmite on. And then when he finished, he just said, "That, Alice, is the reason why there is a channel between England and France." So my French co-workers didn't really like Marmite, but let's see what happens if we take a piece of toast that Liz has prepared earlier and we spread some Vegemite on this toast. Looks pretty black and normal. Let's see what happens if we shine a UV light on there. Spread it a little bit more thin. I've put it on too thick, which is also something I've been accused of at home. Working too well. Maybe I should. Can you see the yellow fluorescence? Yeah. That's the Vegemite. So in the parts where we can't see the yellow fluorescence, it's because it's too thick and it's obscured by, the sticky black goodness of Vegemite. But where it's thinner, we can see this fluorescing. And that's a good way to check that there is enough of one of the molecules that makes Vegemite good for our diet, which is vitamin B12. And this is the molecule that fluoresces inside our Vegemite. So there are also creatures that fluoresce. It's not just molecules it's not just foods, it's not just molecules that we can make in a lab. This is a scorpion that fluoresces under UV light. And why it does this is something that scientists are still trying to work out. But if you shine a UV light in a dark cave, this is a good way to find if there are any scorpions present and to avoid being stung by one. Other animals also fluoresce. This is a beautiful jellyfish that fluoresces blue. And in this process, what's happening is it's the same thing that's happening. There's some light that's being put into these systems in the form of energy. And then when this energy rises up to an excited state and falls back down to the ground state, it's released in the form of light. So we've seen how these beautiful creatures can shine up, light up in the dark, and fluoresce so that we can detect them. Now Liz is going to share what she does with her wonderful research group, which is something that I think is even cooler. Liz and her research team are working on chemistry that literally lights up our cells.
So let's start by thinking about cells. Often when we think about cells or we see pictures of cells, we see pictures that look like this. So they're just empty bags containing water and a few organelles here and there. But that's not at all what a cell looks like. The cell is full of DNA, of lipids, of proteins of all shapes and sizes. And as well as that, it contains many smaller molecules like metals or other small molecules. And so in fact, this is what a cell looks like. It's chock full of a whole lot of molecules, big and small, which are all jostling for space. And in fact, it's pretty hard to move if you're in that environment of the cell. So this makes some problems if we want to understand what's going on inside our cells. If we want to discover where a drug molecule is going and how it's having activity, or if we want to discover a molecule that's causing toxicity and where it goes, or if we want to discover new molecules that have never been seen before. We've got this really complicated picture, and it's very hard to pick out one part of the cell from another. So one way that we can make things less complicated for ourselves is to turn out the lights, to get rid of all of this complexity, and instead we can just light up parts of the cell, whether it's the membrane of the cell, or the nucleus, or the mitochondria. And we can even use this to light up new molecules in the cell that we've never really seen before. We've already seen how useful fluorescence is because it lets us see things really clearly in the dark. And so our strategy is to use fluorescent molecules to do this lighting up of different parts of the cell. If we think about this in another way, we can think about where's Wally? And now some of you may have pored over these pictures in the past, and I'm sure many of you can immediately find where Wally is. But imagine how much easier things would be if we gave Wally a torch. We could then turn out the lights, let Wally turn on his torch, and immediately, very quickly, with no effort, we would see where he was. And this is something that we can do for molecules. We can give them a torch, or in fact give them a fluorescent molecule, and let them signal where they are. And this is particularly important for drug molecules. If we can label them with a fluorescent molecule, we can then see where the drug molecule is within cells or within the body. So one thing that we've done in our lab is to use this strategy to understand cisplatin. Cisplatin is a really important anti-cancer drug. It's been used for a very long time, about 40 years in the clinic, and it continues to be one of the most important anti-cancer drugs. It's used in over half of all chemotherapies within Australia. Despite that, though, it has many severe side effects, and so we really need to understand it better. We need to see where it's going within cells and where it's going within the body. So in order to do this, we've taken cisplatin and we've labeled it with a fluorescent molecule. We've given it a torch so that it can tell us where it is in the body and where it is inside cells. And so now we can start to understand it better and hopefully understand how we can improve the drug and make it a better drug with less side effects. So fluorescence is really useful at telling us about the location of molecules, particularly drug molecules in the cell. But we may also want to be able to discover where there are chemicals in different environments. And so to give you a very simple example of this, we can think about how tonic water is useful about reporting on chemical environments. So here we had our tonic water. Thanks. Here we had our tonic water before. Remember, it is highly fluorescent. I'm now going to put in some salt. This is just regular table salt.
And try this at home if you've got a UV lamp.
And if you don't mind it bubbling over. And now we no longer see the fluorescence of the quinine in the tonic water. And the reason for that is that salt quenches the fluorescence of the quinine. We can think about this in another way. We can think about the quinine as a sensor for salt. If we want to know if there is salt present or not, we can put tonic water in and see whether we see fluorescence.
So I prepared 2 drinks for Liz earlier, one that was made of seawater and one that's made of tap water, because we like to play tricks in the chemistry department. But Liz has the skills she needs to tell these apart. You can see if you look at both of these, they're just 2 colourless samples. And if we place those on the UV lamp, you can see that they glow slightly. But we can now detect which one is safe for Liz to drink by adding a little bit of tonic water. So we can see that one of these is glowing much more brightly, which means that this is the tap water. There's no salt in this sample, so the fluorescence isn't quenched, whereas this one, the seawater, has quite a bit of salt, and it's quenching the fluorescence of the quinine.
So we saw that quinine is a really easy example of a sensor, but we don't want to measure salt in our bodies. There's salt everywhere in our bodies. It's not something that we're particularly interested in. But we can use exactly the same process and the same line of thinking to sense the sorts of molecules that we are interested in studying. And one thing that we've already heard about already tonight that is of very great importance for human health is iron. So we tend to think about iron as something that is used in making nails, and it is actually the most widely used metal in all industrial applications, but it's also the most important transition metal in our bodies. We have about 4 to 5 grams of iron in the average adult body, and that iron plays many important roles. Alice already told us about how iron is important in hemoglobin, in blood, and its most important role in the body is in the transport of oxygen from our lungs to all of the other tissue in our bodies where it is needed. Now, because iron is so important, not just for humans, but for every other organism, it's quite common to not have sufficiently high iron levels and to need to take iron supplements. And the reason for this is that there's an age-old struggle between all organisms for iron. Every organism needs iron in order to survive, and because there is only a limited amount of bioavailable iron, there is a struggle to get the iron, for each organism to get the iron that they need. Now, in my case, the organism that needs my iron is my baby, and so I need to take iron tablets to make sure that there is enough iron for both of us. But many organisms that we don't want in our bodies also fight us for iron. A prime example of that are bacteria.
Yeah, and I take iron supplements because I am low in iron. I'm not very good at absorbing iron. And a few weeks ago, when I wasn't feeling too well and I was taking some antibiotics, Liz told me to stop taking the iron supplements because the bacteria are also fighting for this iron, and maybe I was feeding them by taking this iron supplement. So thanks for that advice, Liz.
So another type of living cell that needs iron are cancer cells. They need iron to grow, and in fact in our bodies they're growing much more rapidly than healthy cells. And so recently a very popular area of research is to see whether we can starve cancer cells of iron as an effective way of killing those cells. If we can stop them from acquiring the iron that they need, then maybe they will fail to survive, and that's a very easy way to kill these cells. This is called iron chelation therapy. The chelation tells us that we're pulling away the iron from the cancer cells. So if we want to understand iron chelation therapy, we need to be able to see if it's working. We need to see if we've successfully changed the iron levels within cells and where in the body it is having greatest effect. And so we've been very interested in making a fluorescent molecule that can act as a sensor for iron in a very similar way that quinine acts as a sensor for salt. So we've made a molecule that looks like this. It contains two fluorescent molecules. It contains this blue fluorescent molecule and a yellow fluorescent molecule, which is actually related to this fluorescein, this molecule. And we find that in the absence of iron, we see both blue and yellow fluorescence, but the iron quenches the fluorescence of our yellow molecule, and so in the presence of iron, we only see the blue fluorescence. So now we can measure blue and yellow fluorescence and use that to tell us how much iron there is. So down the bottom here, you can see some pictures of cells that we have treated with our fluorescent sensor. On the left are control cells not treated with any iron. We can see both blue and yellow fluorescence. When we starve the cells of iron, we see that the yellow fluorescence gets brighter because we're going back to that case on the left where we don't have iron present. And when we feed the cells with iron, we see less yellow fluorescence because now the iron is quenching that yellow fluorophore. And so this can be really useful, and we're starting to use it to understand iron chelation therapy and how effective it is in the body. Another question that we're very interested in, another thing that we would like to sense, is hypoxia. So hypoxia means lack of oxygen. It's the opposite of normoxia, which means a healthy level of oxygen. And we've already said that oxygen is essential. Iron plays a really important role in taking the oxygen where it's needed in tissue. So any tissue in our body that doesn't have enough oxygen, that is hypoxic, will tend to be unhealthy. So hypoxia can occur in stroke, where we don't have enough oxygenated blood to the brain. It can occur in heart attack, where we don't have enough oxygenated blood to the heart. And it can also occur in cancer, where cells grow far away from the blood vessels and they therefore become hypoxic. So we actually want ways to treat the hypoxic cells in cancer. They change their properties and they become harder to kill. But we also want to be able to see where there is hypoxia in healthy organs so that we can treat the effects of heart attack and stroke, for example. So again, we're very interested in using fluorescent molecules to study this, and we've made a fluorescent sensor that tells us about oxygen levels in the body. So we actually made this sensor using molecules that were inspired by nature. This sensor has two fluorescent molecules again. It has the fluorescent molecule on the left is called a coumarin, and it's the same class of fluorescent molecules that make scorpions glow. It's also the same class of molecules that are used in laundry brighteners, the reason that white clothes fluoresce under UV light. The fluorescent molecule on the right is riboflavin, which Alice showed us before, fluoresces yellow, and it's the molecule that makes Vegemite fluoresce. So in healthy tissue, when we have normoxia, we have fluorescence from our riboflavin, so the molecule looks green. In hypoxia, instead, we get a switching off of the green fluorescence and a switching on of the blue fluorescence. So we get this change from the green, which tells us that we're in normoxia, to the blue, which tells us that we're in hypoxia. And so we can use this, and we and many collaborators have used this molecule in many different types of animals, different types of cells, to understand levels of oxygen and how they vary with disease. So we've seen how we can use the small molecules that cause fluorescence in biology. But I'm going to talk now about how we can use the larger molecules that come from organisms. So Alice told us about this beautiful blue jellyfish. It's found off the coast of Africa, and it was discovered in about the 1960s. And very soon after, scientists in Japan discovered that one of the molecules that causes its fluorescence is not a small molecule like the ones we've been showing you, but instead is a protein. And it's a protein called the green fluorescent protein. Now, it's a little bit confusing that the green fluorescent protein should make a jellyfish fluoresce blue, and that's because there are many other proteins involved in many other processes. But if we take out this protein and look at it just on its own, it will fluoresce green. So this molecule is called GFP. Soon after that, some other scientists discovered that they could take the green fluorescent protein and express it in any other animal, and those animals would then fluoresce. So here you can see we've got some fluorescent fish. Now, only some of these fish are fluorescing green, others are fluorescing other colours, and that's because another scientist discovered that he could take the green fluorescent protein and change its colour by just making very simple mutations. So now we have a yellow fluorescent protein and a red fluorescent protein, and we have this beautiful array of differently fluorescing fish. So these three scientists actually won the Nobel Prize for Chemistry in 2008 for these discoveries. So Osamu Shimomura for first isolating the green fluorescent protein, and Martin Chalfie for then discovering that he could put it into any organism, and then Roger Chen for discovering that he could change the colour of the green fluorescent protein. Now, you might question why being able to make coloured fish is such a profound scientific discovery as to win the Nobel Prize. But in fact, this is a scientific discovery that underpins a huge amount of biological research today. And I'm just going to tell you a story about one organism, a very simple organism, where the green fluorescent protein and its other coloured partners have been really important. So this organism here is the C. elegans, or nematode. It's a microscopic worm which is found all around the world in soils. In some cases it can cause disease, in other cases it's perfectly harmless. And it's a very, very important organism for biological studies. It's one of the simplest organisms that has a nervous system, so if we want to understand how our brains operate, we can use a much simpler model to watch and see how signals are transferred from one nerve to another. It's also a really simple system. It has 1,000 cells and they're mapped out quite well across the worm. And finally, it's really useful because it has a lifespan of 2 to 3 weeks, and so it's a very good model for aging. You don't have to wait too long for it to age in order to understand and study the whole life cycle of these worms. So we know that there are 1,000 cells and we've mapped them out and we know what each of them does, but we can't see them very well. If we look under this light microscope image, we can see these are the egg cells ready to hatch or ready to be born, but we can't tell the difference between other cells just under a light microscope. And this is where the green fluorescent protein comes in, because we can selectively express the green fluorescent protein in different types of cells of this worm. So we can express it so it tells us where all of the nerve cells are, or where all of the muscle cells in the body wall are, or where a particular type of nerve cell, these are dopamine responsive nerve cells, exist. We can also use the fact that we can make different colour fluorescent proteins and so we can use two fluorescent proteins at the same time. So here you can see in red, we're looking at a protein that is involved in the breakdown of fat, and in green, we're looking at the proteins that package DNA. And so now we can compare where they are and see where in the body one protein is and not the other.
Well, Liz, I'm going to see your worm and your fish, and I'm going to raise you. Does anybody here have a cat? Anyone here like cats? Anyone have a problem trying to get their cat to come in at night or to find their cat? Well-behaved cats? Well, I know when I had a cat, they used to go missing quite frequently. Well, it turns out what you need is a biochemist who can express the red fluorescent protein into your cat before it's born, and you need a green light, or you need a white cat. So these are two cats that have been shown. There's a green light being shown on these two cats. The one on the right is a white cat that's showing the green colour, it's under a green light, and the one on the left has the red fluorescent protein expressed into each of its cells of the body. So it glows red in green. And what this proves is probably not a viable way to track our pets safely. But the really important part about this research is that it showed that you could express a fluorescent protein into each safely. And as Liz mentioned, this has given us, the world of science, a whole lot of things that we can and discover and actually be able to see these things happening. So the properties of fluorescence and the properties of light and using these molecules to light up biology are already being used in clinical applications. So for those of you who've ever worn contact lenses, you might have had a similar experience to me. Go to the optician, and the optician decides to look at the safety of your eye and detect whether there are scratches on the surface of the eye, which is caused by putting your contact lens. What they use in this case is actually Fluorescein, a few drops of the Fluorescein that we showed you for the rivers, your eye, and they can look at it. Now they don't shine UV light into your eye because that would be quite dangerous, but it fluoresces green and it can show if there are any scratches on the surface of the eye. It's a little bit embarrassing if you have this and you have a cold at the time, because for a few hours afterwards, if you blow your nose, you have some fluorescent markers on the tissue.
So fluorescein is really helpful in telling us about the structure of the eye, and it can give us information like this. It can tell us about, say, a hemorrhage in the eye. It lets us look at the structure of blood vessels within the eye. But we can also use fluorescence to allow us to study disease. It can act as a sensor of disease in the eye. And this is particularly important for understanding dementia and Alzheimer's. So dementia affects about 300,000 Australians, and a large proportion of those have Alzheimer's disease. Alzheimer's disease is characterised by a clumping of proteins in the brain, and so quite commonly it is diagnosed by observing those protein clumps. Now you can imagine that it's pretty impossible in a living person to look at protein clumps that might exist in the brain, and it really can only be done post-mortem. But recently, it's been discovered that some of those protein clumps can also be seen in the eye. And we know that we can actually look at the eye. The eye is close to the edge of our body, and we can use light to look at what's going on within the eye. So a really exciting recent breakthrough was the discovery that curcumin, the molecule in turmeric, actually binds selectively to the protein clumps. And if there are enough of them in the eye, that is, even in early Alzheimer's, we can then look and see the fluorescence of the curcumin and then maybe use it as an early diagnosis of Alzheimer's disease. So this is something that is still in early clinical trials, but it may really let us revolutionize understanding of this disease. So we talked earlier about cancer treatment, and we talked about cisplatin as a useful chemotherapeutic. But another very common form of cancer treatment is surgery, is trying to cut out the tumour so that it is no longer in the body in the place where it is having its detrimental effects. So many of you may remember playing this game, Operation. The idea of the game Operation is to have a steady hand and to pick out all of these strange objects without setting off the alarm, without setting off the electric circuit which sets off the alarm. And sometimes this is something that we think of what surgeons do. We think that all that matters is that they have a steady hand and that they just pluck out the tumour from where it is in the body. But that is not in fact the case. Now I'm going to show you a picture of inside the body, so if you feel like you might be squeamish, you might want to look away briefly. So rather than surgeons seeing a body that looks like this, in fact, this is what their view generally looks like. This is an ovarian tissue, and in this tissue there is a tumour. But we can't see where the tumour is. We can't tell which tissue is cancerous and which tissue is healthy. And this is a problem because the surgeon wants to cut out all of the cancerous tissue, doesn't want to leave any cancer behind, but doesn't want to cut out any tissue that he doesn't have to. So if the surgeon can avoid cutting out the any healthy tissue, there'll be less side effects on the patient afterwards. So we see the picture is really difficult here. We can't see where the cancer is. But what we could do is selectively light up the cancerous tissue with a fluorescent molecule and then give the surgeon a view that looks like this. So now what the surgeon needs to do is to cut out all of the fluorescent tissue and leave behind any tissue that is non-fluorescent. And now it's really easy to tell if you've got all of the cancer out, and it's really easy to make sure that once you think you've taken all the cancer out, to shine a light on it and see whether any fluorescence remains. Again, this is work that is currently in clinical trials, but a number of molecules have been identified that selectively light up cancerous tissue and not the rest of the tissue. And so this is what the surgeon might see. The surgeon might be looking at the camera, and this is an ultraviolet camera which is lighting up all of the fluorescent tissue. So we've seen some examples of how fluorescence is really useful in different types of clinical applications. Really at the beginning, we're not using fluorescence as much as we could. There are many exciting things that we can continue to do in the future. We've also seen how we in our lab are very interested in making molecules that tell us about important aspects of health and disease, that allow us to identify the molecules that might be used to treat disease or the molecules that are essential for the health of our bodies. But these are just 3 examples, and you can imagine that there are many, many more things that we need to sense. If we come back to our picture of Wally, I feel sorry for everyone else in the picture. We're all so focused on finding Wally, and yet there are many interesting other things that are going on in the picture. So we might want to find the man who's smoking the pipe, or we might want to find the salespeople who are bludging on the job, or we might want to find the lady who's falling over. There are countless interesting stories in this picture and so many things still to uncover. And the same is true of the cell. There are so many new proteins and functions and small molecules that we are yet to uncover. We still don't understand, we still only understand a very tiny amount of the exciting chemistry that's going on within our cells. And so this is really what drives our research. We've really only scratched the surface of what we can sense and what we can understand. And so we're now continuing to make new fluorescent molecules to tell us more about all of the different parts, all of the fascinating chemistry that's going on in these systems. Thank you.
So we have time for questions, and there's going to be some roving mics, I think. Top left there to start off with.
This might be a bit off the track, but I was just wondering, what's the evolutionary advantage of fluorescence in the organism? Like, okay, perhaps mating, seeing each other, mating rituals, but the jellyfish, I understand it doesn't have eyes, so they wouldn't see each other. So what is thought to be the advantage of fluorescence?
Yeah, it's a question that hasn't really been resolved completely. I'll answer the scorpion question first, which you didn't ask. So in terms of scorpions, it's thought that in the bright daylight, it actually helps them camouflage, because under the sun, they will fluoresce so bright, well, there will be bright fluorescence that lets them blend in with the other things that are bright around them. It may also be an evolution to help them be caught by other organisms, so it might actually be not advantageous for them, but advantageous for other things that want to catch and eat, particularly the jellyfish.
And if you think about both of those creatures, so the scorpion that lives in a very dark cave, lots of the time, scurrying through these great caves, and jellyfish that live very deep down in the depths of the ocean at times where it's very dark. It could be, some researchers think it's to warn off predators, and similarly could be to attract things that they might want to eat. So I think there's still a lot of research happening in that space, and it's always interesting to find out.
So with glowworms, it's certainly that they attract insects towards them because of the light that they glow, and it could be the same for scorpions, really.
Next question.
A beautiful presentation of the light in today's science. In case of the surgery and cancer, glowing cancer cells, how is the fluorescent molecule, reaching those cancer cells? Are they sprayed? Are they fed? How are they applied?
So it depends what type of cancer is being looked at. In a colonoscopy, say, it will be in the preparation for the same as when you have an enema and you get flushed out with the flush, the dye can be put into the enema, but it can just be sprayed on. Yeah.
Next question over there.
I've been reading recently about them using light in drug delivery. So they've been I can't remember the specifics of it. I think they deliver a molecule inside to where the target is, and then they have another molecule that when it gives off light, it then releases the drug. Can you tell us anything about that? It's a fascinating idea.
Yeah, so look, one of the challenges now in drug discovery is to make drugs that are not toxic until they're at the site where they should have activity, and then to activate them. And so light is a really useful way of activating them, because we can use light in the same way that we use radiation. We can shine a beam of light exactly on where the tumor is. And so the challenge then is to make molecules that we call caged, so they're not active until they are uncaged by light, and then they become active. And then the other type of therapy that's related to that is examples where we shine light on a molecule and it actually generates reactive species, so species like hydrogen peroxide, which are damaging to their environment. So that's something that's actually more clinically advanced than just using light alone.
There's lots of researchers here at the University of Sydney who are looking at these kind of targeted drug delivery systems, again, for the reasons that Liz mentioned. We know that lots of the side effects of chemotherapy, for example, are because chemotherapy kills healthy cells as well as cancer cells, particularly fast-growing healthy cells. But you can use light, and you can also use other things. So you can design cages that open up in a more acidic environment and deliver a medicine, or that open up in the presence of other molecules inside the body. So it's something that a lot of researchers are looking at, and certainly lots of our colleagues here at Sydney are doing really cool research in that area.
Next question.
Elizabeth Vanalis, first of all, thank you very much for your presentation. And on a more general note, thanks very much to the university and the Department of Physics for putting these on every year. They are just wonderful. We've been coming now for about 5 years, and we never cease to be amazed and informed. So thank you very much. But my question is also evolutionary bent a little bit too, because it's interesting how to think that as a species, we've developed in such a way that we only have to survive and thrive by seeing a very small, minute part of the total electromagnetic spectrum. I wonder what we would see. What would we see if it was, say, if we had evolved to double that size? I mean, we'd be seeing what you are now demonstrating to us through your chemical analysis.
I think we'd be even more distracted than we are by our mobile phones. We'd be seeing missing too much information. So, for example, if we looked at the night sky, it wouldn't look dark. We would see different wavelengths of light, so it would be hard to rest, it would be hard to sleep, to get those much needed hours of sleep where we kind of rejuvenate in darkness. But there are many species that can see beyond that, that visible region. So some insects can see in the UV region, and this helps them to find the flowers that they're looking for in order to pollinate. So it's kind of it kind of blows my mind to think about this almost, light is the fastest messenger that we have, right? We can detect things very quickly using light because it travels so quickly. And there are all these messages that are going on at the same time that we're completely unaware of, just because our eyes can't detect that particular wavelength of light.
Next question.
Does the fluorescence of fluorescein degrade over time?
Only if it is always being irradiated with a very, very strong light. So we could have this, if we left this solution here for days and weeks, it can stay the same. But in experiments where we use high energy lasers, we do see a decrease in the fluorescence over time.
In the same way that some of the reactants that I brought into this demonstration, you might have noticed they were in dark bottles. So light can cause lots of reactions to happen. So some molecules are not stable to visible light that can cause other processes. And in a similar way, if you shine UV light on something that fluoresces, it starts to kind of decompose.
One final question, just up the back there, middle. Put your hand up really high. There we go. Can you see him now? He's coming behind you.
Thanks very much for this. What are the experiments using the high-powered lasers?
What are they? So even when we do use microscopes, we use a laser that is shone. So it's not high power, but it's very directed. And so at the specific spot that we're looking at, we will be causing an increase in the energy. And so that is what can cause bleaching of our fluorescence. So that's what we use. We've only talked about biological applications, so we don't use such high energy lasers. We just use highly specific lasers so that they're just focusing on one spot in the cell.
Our friends in physics tend to use those kind of high-powered lasers a bit more, and one of our colleagues, Girish Lakwani, who's just here in the audience, he's also somebody who uses a lot of lasers in his research, and hopefully he will share a Sydney Science Forum at some point in the future. So I've dropped you in there, Girish.
Liz, Alice, it just leads me to do two things. Firstly, as a Scotsman, I've rarely eaten a vegetable, so I'll take the Vegemite home tonight. And secondly, thank you for a really wonderful talk. It's done a couple of things for me. It's reminded me how great the science we do at Sydney University, and that two of our brightest exponents of education of science and the activities and research are in front of us today. Thank you so much. As you walk out the door, you have these feedback cards which have very happy faces on them. Could you take a moment, fill in the cards? It helps the Sydney Science Forum in their feedback. Thank you so much.